So we have completed our lecture till this slide. Okay. So next we will move to the next what I told you about the types of microscope based on the contrast feature. Okay. Is it audible to everybody?
Yes, sir.
Okay. So in the previous class, we were discussing about different types of microscopes. Okay. So different types of microscopes based on their contrast feature today we will discuss. That is how the samples are viewed, how the samples can be seen in our microscope. So whether that sample is having the feature or the capacity to absorb the light within itself and transmit that light, or whether that sample is having the affinity to capture the light and deflect it or reflect it. So based on that, basically we have 4 different types of microscopes. Okay. So what are those 4 different types of microscope? First is your bright field microscopy. Then we have dark field microscopy. Then we have phase contrast microscopy and fluorescence microscopy. Okay. So first let us discuss about what is a bright field microscopy. So as the name says, a bright field. So that means a bright field microscopy is the conventional technique. It is suitable for observing the natural colors of specimen on the observation of a stained sample. The specimen appears darker on the bright background. Okay. So what it is saying is, suppose like we have any sample that you want to see in the uh, under the microscope. So whether that sample, as you can see in this figure, so suppose this is the objective lens of your microscope, and this is the sample which you are going to see, and this is the stage. Okay, so on the stage you have placed the sample, and through that sample the light is projected. Okay, so whether that sample, as you can see the light here, it can pass through the sample. Okay, so if the light is passing through the sample and we are getting the image through the objective lens, so that means the type of microscope that we are using is a bright field microscope. That means there is a bright background, and when that bright light is passing through the sample, so as a result we are able to see that specimen in dark color, and as a result that specimen image is viewed, is seen through the objective lens. Okay, so that is the idea about your bright field microscopy. That means the light can pass through the sample. That is one thing. Then next is about your dark field microscopy. So what does the dark field microscopy say is, a dark field microscopy shows the specimens bright on the dark background. So in comparison, when it was a bright field microscopy, the background was bright. When it is a dark field microscopy, the background is dark. So keep that thing in idea. So in that dark field microscopy, bright field microscope that have a condenser with a filter holder can be easily converted to dark field by placing a patch stop filter into the filter holder. What it is saying is, you can convert your bright field microscope into a dark field microscope also by placing the filter. So the specimens appear bright because they reflect the light from the microscope into the objective. Okay, so this is what it is saying. And in the diagram you can see like the light which is passing through the sample. Okay, so all the light is not passing in through the sample. Some of the light is also deflected. Okay, so it is not completely passing through the sample. Some of the light is also deflected by the sample. So as a result, what we can say is the image formed will be like bright image of that particular sample in the background, which is a dark.
Really?
So the images, as you can see, like when you see through the microscope, which is a bright field microscope, your images will appear somewhat like this. Because as you can see, this is your sample that you are seeing through the microscope, and in the background is bright. Okay, so we can say that this sample might have been seen in the bright field microscopy. In this dark field microscopy, if you see the background is dark in color, and the sample is somewhat deflecting the light. It is not completely absorbing the light. Okay. So as a result, what is happening is the image that is formed, it is on the dark background. So along with this bright field microscopy and dark field microscopy also comes your phase contrast and fluorescence microscopy. So that will take into the next slide. So what is phase contrast? Okay, so microscopy requires a special phase contrast objectives and a special phase contrast condenser. This technique is useful for observing unstained specimens that lack color, example bacteria or some glass fragments also. So when your sample is not able to absorb the stained material, when they cannot hold the staining, and when the samples are somewhat like transparent in nature. Okay, so in that case, we can use this type of microscope, that is your phase contrast microscopy. So in this, what happens is the optics will convert the difference in refractive index of the specimen into the brightness difference. So this is your actual idea. So this is the idea of your phase contrast. So what does that phase contrast do? So when the light is passing through the specimen, okay, so based on its refractive index, the specimen can be seen through the objective lens. Okay, so we are seeing the specimen because there is a change in the refractive index of that sample. So once the light passes through the aperture, so if the light is falling on this bacteria or on some glass fragments, so obviously there will be some change in its refractive index. So as a result, what will happen is the change in refractive index will make the object appear. It will, as a result, it will make an image. That image we can see in the microscope. So the idea is the optics will convert the difference in refractive index of the specimen into brightness difference. This will cause transparent objects to appear bright or darker than their background. Phase contrast is expensive but sometimes necessary to see certain structures in living and unstained cells. So in the previous slide we can see that this phase contrast. So what it is doing is, if you don't choose like the phase contrast microscope, so as a result, whatever sample you are seeing in the microscope, you will not be able to see, because that it is transparent in nature. Okay, so once you use this phase contrast medium, so what will happen is the light which is coming through the aperture and while it is entering into this medium of your specimen, so because there will be a change in the refractive index, and that change in the refractive index will be captured by our microscope. And as a result, the captured refractive index will make an image, and that image we can see through the microscope. So this is the principle about your phase contrast microscope and how we see the sample in the phase contrast microscope. Then comes the fluorescence microscopy. As the name says, the samples that we see will fluorescence. So fluorescence microscopy produces a bright specimen on dark background. The fluorescence microscope is an optical microscope that uses fluorescence instead of or in addition to scattering, reflection, and attenuation or absorption to study the properties of organic or inorganic substances. That means if you see this picture, so in your fluorescence microscope, so only those subjects or only those specimens you will be able to see which has the property to fluorescence. Okay, that is one feature, and that is important feature because any subject or any sample that is not having the property to fluorescence, if you subject that into the fluorescence microscopy, you will not be able to see the image because any sample which has the property to shine in the dark background, that kind of specimen only will be able to see in the fluorescence microscopy. Okay, so what it is saying is all the specimens when viewed in the fluorescence microscopy, they will show a bright in color and the background will be dark. So in the dark background, a bright specimen is seen. Okay, so that specimen, how it is absorbing, how it is fluorescence is when, so the idea of fluorescence, how does any sample fluorescence is when you are subjecting the light on it. So what it will do is it will absorb the light and after some time it is going to release the same amount of light. So, but once the sample so the idea of fluorescence is suppose on the stage you have kept one sample and that sample, if it is not having the property to shine, if it is not having a property to fluorescence, then there is no use of taking the sample in the fluorescence microscopy. But if the property of that sample is to shine in the light, so we can use that sample into our fluorescence microscopy. So what is actually a fluorescence? Any material when the light is subjected on it, when the light is projected on it, so that subject will or that sample will absorb the light that is falling on its surface, and the same amount of light will again be reflected out. It is being emitted out. It is not reflected, but it is emitted out. So that amount of light which is being captured and again when it is given back. So are you able to hear me?
Yes, sir.
Fine. Okay. So the amount of light that is being captured and again which is being released. So there will be a small fraction of time. So that change in the time will result into its glowing in nature. The specimen is going to shine in that area. So the change when the light is being absorbed and when it is being given back. So during this process, it will take a small time. So within that short period of time, when the light is being given back, the change in the wavelength of that light will result into its fluorescence nature. Okay, so whatever sample we are placing into the fluorescence microscope, so when we are observing the specimen, the image that we get into the fluorescence microscope will be little bit delayed. Okay. So at once, like once you place the light, once you place the subject and focus your light, instantly you cannot get the result, or instantly the image will not be formed. So it will not take so much of time, but a fraction of time it will take, and the image will slowly develop. Okay, so the idea is capturing the light, emitting the light, and between that, the process of fluorescence will happen, and that thing is being recorded into the microscope. Okay, so that is all about your four different types of microscopes based on its contrast feature. So contrast feature will show you whether the microscope is bright in field, dark in field, whether it is a phase contrast or fluorescence. Okay. So next we come to the introduction of your compound microscope. Okay. So compound microscope will be somewhat similar to your stereo microscope, but there is a small difference. Okay. So if you closely observe this microscope, this same structure of microscope is present in our lab also. Okay. So if you see that in the introduction class for your microscope, I told you that the stage can also, the sample stage where the sample is kept, this stage that can be moved also. Okay. So to move this stage left or right, or forward and backward, these two knobs are there. That is your stage controls. So with these two knobs, you can either move your stage backward, forward, or left and right. Okay. So almost all the components are somewhat same. Their function is also same. Okay. So it is your this eyepiece lens, then you have your objective lens, there is a nose piece. With the help of this nose piece, you can just rotate and fix your desired. Are you able to hear me? Yes, sir? Hello? Hello?
Sir, not
Oh,
audible.
okay, wait. So from this slide, I'll just re-continue. Okay. So the introduction, I will tell you. As you can see, like this microscope, it is somewhat same like your stereo microscope. The only difference is the area. Okay. And one more thing is like the stage. The stage is rotate that it is subjected to your movement. So you can either move your this stage left or right, forward and backward. That is one thing. Then again, one more thing is the objective lens. In this compound microscope, we have the choice of choosing the objective lens. Okay, so compound microscope, what is there? The parts is somewhat similar of what we have seen in the introduction class. There is a eyepiece, there is a head, there is an arm which we use to hold the microscope. Then we have a nose piece on which the objective lenses are fixed. We can rotate it according to our desired thing. Then we have the mechanical stage, coarse and fine adjustment knob is there. Then stage control is there for stage. Then we have the base. And then one more thing I was I will tell you the brightness adjustment. That is one new thing that has been added. It was there in the introduction microscope also, but it was not shown there. So this thing is your brightness adjustment. That is, once you switch on the light, so this halogen lamp is going to glow. Okay. So how much it is glowing or how much the light is being projected or given by this light source, that amount of light source we can adjust with the help of this brightness adjustment. So if we want a small amount of light, so we will adjust that thing. If we want a bright light, we can adjust with this thing. Okay, so the brightness of this can be adjusted. So again, we have the condenser is there, diaphragm is there, aperture is there, stage clip is there. So almost everything you have already gone through the introduction class. Okay, so we'll come to little theory portion of this compound microscope. It is designed for viewing specimens at the cellular level on glass slide. Okay, so cellular level means microscopic specimens which are not seen through the naked eye. Okay, so compound microscope is made for viewing the sample which are in cellular level, that is microscopic in nature, very small, and it offers a greater magnification power than stereo microscopes. Okay, so in this compound microscope, the magnification power is more. Okay, but they have a much shallower depth of field. Okay, so again, this word is coming, depth of field. So these technical terms you should be very much familiar. Okay, so what the depth of field is saying is within the axis or within the direction that you are seeing the sample is how far you can see the sample, very clearly. That is your depth of field. Okay, so that's why this type of microscope is also known as a flat field microscope. Okay, compound microscopes are also usually equipped with multiple objective lenses that can be rotated to adjust their magnification power. I already told you. So now comes your stereo microscope. As you can see, this stereo microscope as compared to your compound microscope, it is having a very limited feature. That is your only one objective you see. This is important. Okay. This is having only in the nose piece, there is only one objective lens. Okay. The eyepiece is two, but this is your only one objective lens is there. Okay. Then again you have this knob whether to move it upwards or downwards, that is there. Then you have your stage clips is there. Okay. Then this stage is there. And if you observe the diagram, the stage is not like movable. That is one thing. Then again, then very important feature is like the area, the space between your objective lens and your specimen. This length of area, the area between your stage and your this objective lens is always more as compared to your this compound microscope. Okay, when you see this area between your objective lens and your stage, it is very small, and in this the area is large. Okay, and that too the moving stage is not there, option of adjusting the objective lenses, it is not there. Okay, stereo microscope offers this is the only like the advantage of stereo microscope is like if you say that we are liking about the objective lens options and all those things, so the thing is some specimens which needs a greater depth of field. Okay, so that thing can be seen into our stage, and all those specimen which needs a like 3D viewing. Some insects are there, or some grains are there, some concrete material is there, some fiber is there which needs a 3D view. So those kind of samples we can easily see in this stereo microscope. Whereas if you see that thing into the compound microscope, the things will not be visible. Okay. So stereo microscope offers a relatively low magnification power. That is one drawback, and a deep depth of field. So this is your advantage. That is ideal for viewing objects, well suited for viewing relatively large specimens such as insects, fossil, that is fossil, the remains, flowers, mineral specimens. So if you have to see these kind of samples, you will have to use your stereo microscope. If you place this things on the, what you say, in the compound microscope, you will not be able to see. It will be completely black. There will be a uh, dark image form. So you will not be able to identify whether it is an insect, fossil, or flower. Okay. That is one thing. Okay. So I will just talk about this compound microscope, then we can end the class for today. So this is about your comparison microscope. So the idea of this comparison microscope is to compare two identity or two different subjects at the same time. Okay, so in one plane, in the one image, you will be able to see uh, two different specimens, but in the same resulting image. Okay, so the idea is that the comparison microscope, if you split it half, so this will be one full separate microscope, and this will be your full individual microscope. So this is actually the clubbing or mixing of two different compound microscope. They have been joined here with this bridge. Okay, that is the only idea. So two compound microscope, when they are bridged together, and as a result, what we can say is two specimen, one from this place, one from this place, and when you see through this eyepiece, the image that you will be able to see is like this resulting image. Okay, so this image, what will happen is, like, if you see, there is a very fine line in this. So every microscope will have this kind of image. So half of the image will be formed from one microscope, and another half of image will be formed by the other microscope. So that is the idea. So this was actually, just look at the theory portion. The comparison microscope was invented in 1920s. In 1920s by Calvin Goddard. Okay. Are you able to see? Are you able to hear me?
Yes, sir.
Okay. 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 So there will be some little disturbance because I'm getting a call also. We'll just finish this topic and we can end the session. Okay. So the idea is this microscope was developed by American colonel whose name is Calvin Goddard in 1920s. So what he did is he took like two different compound microscope, and he bridged with this bridge, so that once we are seeing two different identities into the same plane, so we can compare two different objectives, two different samples at the same time. So that was the idea. So as a result, in the microscope, a comparison microscope, what you can do is two individual articles you can compare at the same time within one view of focus. Okay, so that was the mechanism developed by your Calvin Goddard. Then there was one Swedish person from Sweden, so Harry Söderman. So what he did is he invented a system for rotating the bullets under the objectives. So previously, if you see like in this microscope, okay, in this microscope, there is no function or there is no option to hold the bullet. Okay, so actually I will show you that microscope some other day. So in that microscope, in that comparison microscope, this particular thing is there, that is uh, the system for rotating the bullets under the objective. So what you can do is you hold the bullet or the cartridge on the stage with the clipping, and you can rotate that bullet or cartridge at the same time, and you can match the scratches or the markings that have been developed on that. Okay, so that is the only addition which Harry Söderman added into the previous comparison microscope, and he gave a different term for that comparison microscope what was already made by Calvin Goddard. The only thing he added is the rotating feature on that stage. So he named his microscope as a hastoscope. Okay. So the comparison microscope is used in forensic science to compare microscopic patterns and identify or deny their common origin. That means the two different identities which we are seeing through the microscope, whether they come from the same source or they are from the different source. So that is the main motive to make this comparison microscope or use the microscope. The optical bridge consists of a series of lens and mirror that brings two images back together at the single eyepiece. That means from image formed from this microscope and this microscope, that will be bridged by some mirror and prism which is located into this bridge area, and as a result the image from half image from this area, half image from this area will be clubbed together and made as one separate image and will be subjected to view in this area. Okay, so the user looks through the eyepiece as with a regular microscope, except that a line in the middle separates the circular view field into two parts, right? So as a result, you will get this kind of image. So it is also possible to superimpose. That means one above the other. So one image that you have formed in this area and one image that you have formed in this area. So we can even superimpose. That means one image over the other also we can keep. Okay. It is also possible to superimpose the view fields generated by the two microscopes. But as I'm telling you, like in the normal microscopes, in the normal comparison microscope, the features are very much limited. As you can see, this is your basic kind of your comparison microscope. Okay, here you are not having a feature to rotate the bullets and cartridges. That is one thing missing. Then again, the feature like superimposing, that means image from one microscope over the other. So those two images developed, they cannot be superimposed in this feature, in this particular comparison microscope. But when you buy a high-end microscope, very sophisticated one, then you have these kind of features. Okay. So this particularly convenient when the forensic scientist compares impressed patterns rather than striated patterns. Okay. So when you have to compare this kind of scratch marks or striated patterns, so we can use this normal comparison microscope. But if we have to use like whether the firing pin, this is the base of your cartridge. So if this is your firing pin, and if you want to compare that this depression, this particular impression that is formed, or the depression that is formed, is whether from the same firearm that has been recovered from the crime scene or from the suspect. So what you do is you do the test fire, you collect the the cartridge, you project one side of your this base on one side, and recovered cartridge, and you view from this side. So the similar kind of pattern, this will be formed here also. So what you do is, if you want to superimpose or bring the image from this area over this, and if it is exactly 100% matching, that means the area, the diameter is exactly matching, and the depression is also matching the same. That means we have the same common origin. So if we have to compare this impression, that is striated, normal comparison microscope we can use. If we have to use the impress pattern, we will need the high-end microscope. Mostly used reflected light setting, but transmitted light setting is also available in some instances, and fluorescence light setting are also found. Okay, so if you go for high-end microscope, comparison microscope, the features of transmitted light also that can be that is you can get. Okay, the use is to identify the tool marks, firearms, paint chips, identification of typewriters. Okay, the time is running out, so I will end my